Welcome to Discover ASAP, the video interview series where we highlight the top findings and tools connected to the ASAP network. Today, I'm talking with Camille Goldman from Team Vangheluwe about genetically encoded and modular subcellular organelle probes, or GEMscope, that they developed. Thank you so much for joining me today, Camille. To start off with, can you briefly tell us about your ASAP project and the impact that you hope to have on PD?
Yeah, so thank you so much, first of all, for inviting me to share about GEMscope. So I'm part of the Impact PD team, and our project is interested in looking at lysosomal dysfunction in Parkinson's disease with a focus on lysosomal lipid and polyamine homeostasis. And so our focus is on the PD-related lysosomal genes GBA1, ATP13A2, and ATP10B. And these are genes that are associated, or proteins that are associated with lipid and polyamine transport and metabolism in the lysosome, and they're all associated with risk for Parkinson's disease. And so we're trying to understand how mutations to these genes affect lysosomal function, and then the downstream effect of that lysosomal disruption on alpha-synuclein aggregation and dopaminergic neuron survival. And so my lab, specifically the Blanchard lab, studies these genes in human stem cell models. But the Impact PD team also uses rodent models and patient samples to answer these questions. And so then our overarching kind of goal is to understand how a pathway like lysosomal disruption that has been widely implicated in Parkinson's disease, both in familial and in idiopathic cases, contributes to the clinical characteristics of Parkinson's disease, with the hope of identifying therapeutic targets that we can take advantage of to develop cures.
Great, that's awesome. So let's just jump right in and talk about what GEMscope is. Can you give us a brief overview?
Yeah, so GEMscope is an acronym, and it stands for Genetically Encoded and Modular Subcellular Organelle Probes. And essentially what it is it's a suite of lentiviral plasmids that encode fluorophores that are localized to specific organelles. So we, in GEMscope so far, we've integrated localization of fluorophores to the nucleus, the cytoplasm, mitochondria, and lysosomes. And then we've also included fluorophores that are both single emission wavelengths or more complicated fluorophores that have dual emissions, such as like some FRET fluorophores that respond differently, have different emissions based on their local environment. And these are all localized specifically either to the mitochondria, the lysosome, the nucleus, or the cytoplasm, depending on the localization sequence that is included in the plasmid. And the whole system is incredibly modular. So the localization sequence and the fluorophore can easily be swapped out to meet the needs of the scientific question that's being asked. And these probes are used to image subcellular structures in live cells over time in response to stress or genetic perturbations. And they can even be multiplexed together, so you can monitor multiple organelles simultaneously in the same cell.
Great, that sounds like such a useful tool to have. And so you've already sort of touched on some of the advantages of GEMscope, but I'd love to hear more from you about why you created this tool and any advantages that it has over other tools that like it that might exist, or maybe there are no other tools like it.
Yeah, so we created GEMscope because we found that there was a limited availability of live cell imaging tools, and we really wanted to be able to image ourselves live as they were responding to different stressors, and not just once we fixed them and stained them with antibodies. And so there are available tools. I won't pretend that they don't exist. Other plasmids have already been published, and there's also dyes like Hoechst for the nuclei, or Lyser tracker and mitotracker for lysosomes and mitochondria, respectively. And I admit that those are incredibly convenient tools, and I've used them in my research myself. But it's really unclear how those sorts of dyes, like the long-term impacts of those dyes are on cells, especially if you're doing long-term live cell imaging. In the manuscript, we even show how Hoechst over time leaches out of the nucleus. And then in terms of you being able to segment the images to identify the nuclei becomes really difficult. But if you localize a fluorophore, specifically in our case with an H2B, a histone 2B localization sequence to the nuclei, the fluorophore never leaves the nucleus. So we're taking advantage of genetic signals to keep our fluorophores exactly in the organelles that we want for the duration of our experiment without concerns about what an exogenous dye might be doing to the behavior of our cells. And then I think the most important thing really to highlight with GemScope is the modularity of it. So I briefly mentioned that some of these sort of probes exist. You can find a GFP localized to the mitochondria already on AdGene. What GemScope is that it's the same backbone and the same kind of structure of sequence. So modular breakdown there's a modular construction of the plasmid that allows us to basically expand GemScope infinitely to meet whatever our needs are. Mm-hmm. So the 4 domains to the coding sequence in a GemScope plasmid is the promoter, the localization sequence, the fluorophore, and the antibiotic selection. And in between each of these modules, as I call them, there's a restriction enzyme site that I designed into it. So if there's a fluorophore you want to integrate in that isn't already available in GemScope, or an organelle that you're particularly interested in that isn't the nucleus, mitochondria, or lysosome, or maybe you want GemScope to only be expressed in certain cells or at certain stages, you can take out any one of these modules and replace it with the fluorophore, antibiotic selection, localization sequence, promoter, etc., that you're interested in for your question. And so you don't have to find or hope that there is something already available. It's very easy to generate exactly the probe you want yourself. So GemScope, as is published, is a finite set of plasmids, but it has the potential to be infinitely large to meet really whatever live imaging question need you have.
Great. That's amazing. That sounds like such a fantastic resource to not have to start sort of from scratch every time you want to create some new live imaging tool. So these tools that these probes that you've generated, I'm imagining require a certain level of you have to get them into the cells that you're interested in or into the organelles that you're interested in. So can you share any tips and tricks for using GemScope probes? Are there certain steps in the protocol that require special attention that you want to sort of point out to our viewers?
Yeah, I'll say overall GemScope is very user-friendly. From a cloning perspective, I will say that we generated most of our plasmids via Gibson cloning. So we know that works very well. But as I mentioned, they can be modified with simple restriction, digest cloning, or really any cloning method of your preference. So you don't have, like in the methods of our paper, we describe how we did the Gibson cloning protocol, but you're actually not restricted to just doing it with that method. So whatever cloning method suits your skill set or that you're comfortable with should work for generating new GemScope probes from the existing ones. The step in the protocol I think that has the most variability or is the most important in terms of generating good images is being able to generate good lentivirus titers and then having a cell type that is will be transduced easily by lentivirus. So when we have low lentivirus quality, very few of the cells get transduced, and then it's few and far between of the cells that actually fluoresce and then therefore can be analyzed for imaging. The higher the quality of the lentivirus production, the easier time we have imaging. And then some of this can be mitigated with the antibiotic selection module. So if you include antibiotic selection and then select the cells before imaging, that can really help reduce that effect. But again, if the transduction is really inefficient and your cells don't really reproduce, that might not be super helpful if you end up killing off 90% of the cells with the antibiotic. So really making sure that you're comfortable making lentivirus and having high quality lentivirus before continuing with the transduction. I think that's probably the single most important step to having this work.
Great. That's really good to know. So if someone else wanted to use these GemScope probes in their lab, how could they access these probes for their own use?
Yeah, so we're making them all available on AdGene. They're still currently being processed on AdGene. I don't know if they'll be available by the time this recording goes live, but as of June 2nd, they're still being processed, but all the paperwork's done. So as soon as AdGene approves them, they should be good to go. There are 11 probes that are actively used in the manuscript, but we actually generated 19 over in the process of getting this up and running. We just didn't end up using them all in what we published in the paper, but we're making all 19 available on AdGene. And so I'm sure it'll be linked in the hub. It's definitely going to be linked in the paper. And so, yeah, we hope that people find them useful.
Great. Yeah, we will link to the paper that GemScope is in the caption below. So if you guys are interested in finding these probes or learning more about them, that will be there for you later. So Camille, I like to end my interview by asking if there's anything else you'd like to share about these probes that I didn't ask you about.
Yeah, I'll just mention just one more thing because we talked really about them as a tool. And so in the paper, we hope everyone who's interested goes and takes a look, but we did demonstrate the usability of these probes to identify disease-related phenotypes. So we used a Parkin knockout astrocyte model of Parkinson's disease and looked at proliferation, changes in mitochondrial turnover, reactive oxygen species, and lysosomal distribution, and found some really interesting, relevant changes to those phenotypes, just to show that these probes, while just pretty, also are able to pick out and resolve changes on the subcellular level that are relevant to Parkinson's disease.
Great. Thank you so much for joining me today.
Of course. Thank you so much for having me.